It's my great pleasure to introduce my dear friend, Alejandro Arias. Alejo, he's from Colombia. He's biologist. He came to Europe. He came to the Netherlands, to Rotterdam, to study genetic epidemiology. He did a PhD in Erasmus MC. We share the same thesis director, Cornelia van Dijn. And this is something really, it's like going to the military service together.
Yeah, something like that.
Something that's really a bond. It's a strong bond. And but then Alejo moved to Radboud University in Nijmegen, where he started working with the psychiatrist.
Yes.
You moved to the other side.
To the
Because he started in neurology. He started doing Alzheimer's disease. He did his PhD on Alzheimer's disease, but then he moved to Radboud University to collaborate with the psychiatrist. And he works first in neuroimaging and genetics, right?
and ADHD first.
ADHD. Okay. And now from 2014, 15,
yeah, around
he
there.
started a new research line on microbiome and psychiatric diseases,
Yes,
mainly
correct.
ADHD. And he's going to tell us a really, really exciting.
Well, I hope it's
Thank you very
exciting.
much. The floor is yours.
Thank you, Pascual. I'm sorry I'm the one standing between you and your food, so I'll try to keep it interesting. I'm gonna touch about a wide variety of topics. So if you have a question, just interrupt me. It's better like that. Um, um, there are some things that I don't expect you to know, so I'm gonna take a little bit more time explaining. So yeah, it might take a bit longer there. The other part of the talk that I'm gonna take some time is talk to you about the results. What do they mean, right? Not so much about the statistics or the lab work, because that's really long, right? So yeah, I work in Nijmegen. That's in the east of the Netherlands. So it's not the cool part of the country, right? So you have Amsterdam and Rotterdam here, and then you have Germany here. Next to Germany, that's where we work. It's a reference hospital in the country. It's one of the biggest psychiatric services in the country that is characterized by working with highly comorbid patients. So people that are really suffering, people that are,
Sí, Clemente,
treatment
que sí.
resistant.
Es que van a
Uh,
salir.
okay.
Vale, pero te he dicho que me he conectado a un seminario y
Anyway, so we have like a very interesting resource of patients. They're it's very competitive. Everybody wants to work with these patients. So I will show very briefly one of the things we can do with this type of population. As Pascual said, my strength, my core business is neurodevelopmental diseases, from which ADHD. So I'm gonna skew my talk a little bit about that. I'll try to explain very briefly what ADHD is and how does it relate to our daily life. Try to demystify ADHD a little bit, show you a little bit what it is, and try to give you a picture of how we do our work. I try to bring my knowledge. I did 20 years of genetic epidemiology, so I'm trying to bring that into this new microbiological dominated world, which in terms of analysis and treating the data and revising hypotheses is still the Wild West. There is a lot to be learned in terms of being, in registering studies and stating hypotheses clearly. And this is very important. Sample size, study design, the basic stuff that you learn in epidemiology class or basic statistics. These things we need to apply better into the microbiota world. Not to say that it's not very cool. It is very cool, especially because these people can discover function, right? And statistics, you cannot do that. You need biology for that. So I'm trying to merge these two things. So I will show you a little bit of the way we do it. So let's see. This one works. Maybe. Now. Anticlimactic. I'm sorry. Okay. Eso, gracias. Okay, so we have to align some concepts because words can be tricky and words are important, especially in science. So we use the word microbiome loosely with the word microbiota, but they mean different things. So I'm not gonna be that guy. I I don't mind that much, but I just want you to learn the difference because it's important. So microbiome work refers more to function and to genomes and to biology, right? It's more applied. It's more in terms of research. It's better to use microbiome, but you can also use microbiota. But microbiota is more ecological. It's more the fact that organisms exist and they occupy a place in the ecosystem. And this doesn't refer only to bacteria. It refers to every microorganism that lives in the soil, in the water, in our bodies, right? So it's extremely fast, very, very big. The things that in science, we need to narrow the problem. Otherwise, we couldn't solve it or investigate it. So you have millions and millions of organisms, then we only look at a fraction of those. So it's important that you recognize the difference. And I try to be very strict in the way I use the words, but I mean, I'm not a microbiologist. So we all make mistakes. But now you know. Now you know the difference. Okay. So raise your hands. Who has felt butterflies in your stomach? Raise your hand. Right? So if you haven't felt that, something is wrong with you. I'm sorry. No, I'm joking. That's not true. But the thing is that there is a very strong connection between our brain and our GI system, gastrointestinal system. And that connection goes beyond the obvious things like, satiety or hunger or infection, right? If we have an infection in our GI tract, we feel it in our head, right? But where do we feel somatically? Where do we feel stress? Where do we feel it? In your gut. Right? So this is a very, very strong, high-speed, bi-directional, super information speed. So think about the internet, but bigger, with more information, working 24/7, always picking up signals and processing them back and forth all the time. We call that the gut-brain axis. And there are multiple axes in our body. You have the hepatic brain axis. You have also axis built through bone and other type of physiology. But the one we're going to study is the gut-brain axis, because that's the one where our gut bacteria exerts a more clear, stronger effect. The gut-brain axis, let's say, includes hardware, which is our nerval communication. So over 500 million neurons innervate our GI tract. So it's a place with the second number of neurons in the body after the brain. So there's a lot of information being picked up, for infection, immune response, et cetera, et cetera. That information through the hardware, through our nerves, moves to the brain. And it innervates the brain, the insula receives the vagus nerve. So we are hardwired with the GI tract, which is pretty cool, right? We also have immune system. So the immune response is not only regulated at the GI level, but also regulated at the brain level. Even though we have our own immune machinery in the brain for, internal immune function, signaling from the brain helps us, boost up our immune response, for example, when we are confronted with stressful situations. Very simple. I'm not going to go into that because immunology is like an occult science. And I don't know enough to tell you anything reasonable about it. So I'm going to stop there before I make a fool of myself. Now, the third thing is the endocrine system. And this is very important for the talk of today, because in our GI tract, we produce a lot of things that we use for our, somatic functioning, but also for our brain function. The cool thing about this is that the bacteria that live in our gut modify all those signals. Very beautiful studies have shown that doing vagotomies in brain in animals, right, modify not only the diversity of the bacteria, right, but changing the landscape of bacteria in these animals and then performing the vagotomy results in different behavioral states. So we also know that the immune system, the first steps, the first blocks of immunity, innate immunity, are given by the bacteria that we all receive from our mothers when we're born. So we are exposed for the first time to not a pathogen, to an external pressure through these bacteria when we're being born, which is pretty amazing. And it continues to work together with our immune system until the day we die. And then thirdly is the endocrine system. Bacteria take the stuff we eat, they break it down, and then they produce substances that we do not produce. And these substances include tyrosine, phenylalanine. These are and other substances, and then they are used to create molecules like serotonin. You've heard of that, right? Serotonin is 90% of serotonin is produced. Systemic serotonin, sorry, is produced by the gut bacteria. They make it. So it not only helps us, move their food around in our bowels, but it also helps us in many other processes, neural communication in the system, et cetera, et cetera. But the key issue here is that they produce these precursors, right? And these precursors can have a potential effect at the brain level. I will explain that. Before I get there, I just wanted to show you with this infographic that very, very briefly shows how complex this is. So I'm talking in very simple terms, trying to give a very clear picture about this relationship and this effect. But we're talking about millions of microorganisms split into thousands and thousands of species with redundant and sometimes conflicting functionality that depend on very specific ecological conditions like pH and temperature and humidity, et cetera, et cetera. I mean, our bowels are a very nice place to live. It's very good housing for these things, and we give them a lot of food, so they make a lot of things for us. Right? This is the perfect one of the nicest examples of coevolution, because these bacteria live among us. They live in our skin, in our throat, in our mucosa, right? But it's not random. We know that it's not random. From all the bacteria that we see in nature, only a selection of those bacteria live with humans. So that means that there is coevolution. So genome determines which and how many of these bacteria live among human population. That's pretty cool. I think that's pretty cool. Now, but the level of complexity is gigantic. So the size, the number of bacteria, the number of cells, right? The number of individuals. I mean, the infographic is fantastic. It was created by the group in Cork in Ireland. These are let's say they are the forefront in the research of microbiota and health in the world. Pretty cool group. And they created this very nice poster that I like to share with people, because it's not as simple as we think. Everything in science and medical or life sciences is way more complicated than we think. Now, if we want to zoom into the microbiota, there are multiple determinants of how it works, right? And I'm just naming some of them, but they are pretty close to us, right? Where do you live? How old you are? If you exercise, etc. And the most important right now identified are your age and what you eat. The other ones modulate, but these two are the ones with the biggest effect in the shortest time. And with age, everything changes from the day you're born until the day you die. Now, the effect of microbiota extend, of course, to normal homeostasis, but also affects disease. Among those related to our central nervous system. And in that sense, the gut-brain axis here, seen to the side, you can look at it from, the more gut side, or you can look at it from more the brain side. In this case, we're going to look at the brain side, how this interaction works. And for the purpose of today, we're going to focus on the production of neurotransmitters. I promise you I was going to talk about that again. It is important because we postulated this a few years ago, and we wanted to see if this still want, because we haven't been able to do it, if the products, the metabolic products of the bacteria through the blood circulatory system can go through the blood-brain barrier and then affect the actual kinetics of brain chemistry, which is pretty important, right? If you think about ADHD, right, which the problem of ADHD, one of the problems is the reuptake of dopamine, right? What happens if we could compensate the amount of dopamine through bacteria? It would mean that this person could present with a milder phenotype, for example, be less impaired by the disease, by the disorder, and have a functional life. The same could be seen for depression in case of serotonin levels and other hormones at the brain. Now, the opposite can also be true. Perhaps we miss the right amount of bacteria or the right type of bacteria, and we don't have enough. So on one hand, we could compensate, a bad quote unquote genome. Please don't quote me on that. Or the other thing is that by not having the right amount of bacteria, perhaps we're going to miss out and be at increased risk of some neurodevelopmental disorder. So this whole idea is so ideally where we want to reach in our work, and we postulated this a few years ago. And while we're very busy here, somewhat busy here, this connection we still haven't been able to describe it. It's quite difficult actually to do. Now, so how does this come together? Well, we know now that there are specific things happening at a specific developmental ages, and when we're born, things start to happen very fast at different levels: genetic, neurological, immunological. Right? There is a lot of developments that affect the actual processes early in life. This has been well documented, and they include, in utero effects, includes diet, include disease, and these things have different effects at different parts of the body. Now, why is this important? Well, it is important because the onset of neurodevelopmental diseases is at early age. I mean, and by onset, you need to understand that this is how we can properly measure it. It's not that it's not there, it's that our tools that we have to diagnose a child with autism or ADHD can only happen after a given number of years. I think it's 6 years for autism. I think it's at least 7 years for ADHD. But those effects are happening earlier in life, even in utero effects, perinatal effects, etc., etc. So having all this information in hand and assuming that the microbiota plays a role, hopefully I convince you a little bit about that, then we technically could use the microbiota to influence that risk. Technically, that is the premise that I'm putting out there, right? So that means that if we can modify the bacteria, we could modify the risk. And there is one thing that we can modify is the bacteria, because I told you already that you can modify it with diet, you can modify it with lifestyle factors, you can modify it with other things. It's not like genes, right? Genes are more static and not completely fixed, but are way more static. Okay? So this is where ADHD plays a role. The Spanish translation is TDAH, trastorno de déficit de atención y hiperactividad. And it's very frequent in children, right? And can persist into adulthood. So between 2.5% and 5% in children worldwide present with ADHD. Half of those children will persist into adulthood. So it's pretty frequent, actually. The thing is that I'm not going to go into too much detail, but very important for you guys to know is that it's a genetic multifactorial disease. The heritability have been estimated very, very consistently in the last 50 years, and it's around 75%. This is one of the, let's say, success stories in genetics. It's very stable. Medication works very well. It's the best one in the market, around 70% of response rate. Of course, with side effects and all that stuff, and many societal problems that I will explain later. But it's very robust. The genetics are very well established. The clinics are also very well established. And now we have data to do GWAS. I think you've heard something about GWAS here, right? I know Pascual likes the GWAS a lot. So we also have GWAS or ADHD. And the numbers are not nothing, eh? This is not like bone or height or anything like that. But I mean, it's pretty decent numbers, closely, over 200,000 people, and pretty robust peaks that keep up coming and coming and coming. One thing you don't see from those peaks are the dopamine genes, are the GABA genes, the serotonin genes. They're not coming up here. So those results somehow reinforce, or maybe I'm just too biased, reinforce this compensation hypothesis that I'm trying to sell you on, that if you have the good bacteria, you're not going to see the dopamine problems. So that's why the dopamine genes might not appear, because not everybody with ADHD becomes a case, because the bacteria are compensating it. And that's why we don't see them here. That's just me wishful thinking. As I told you, the treatment is very good, right? What we're trying to determine is that how diet and other lifestyle factors affect, because people with ADHD behave differently. And that means that they eat differently. That means that they move differently. They could just leave it there, because otherwise I can talk about this for hours. So, but there is a distinct pattern in other ways of measuring behavior that go beyond the clinical, the DSM, let's say, rules, which include dietary behavior and lifestyle. Okay, keep that in mind. Now, right now, I think I've convinced you that modifying diet has an effect on bacteria. This very strong relationship. Now, the other thing I want to make a point on is that diversity of nutritional behavior might have or can have an effect on ADHD. The strength of this relationship is not that good, but there is data, and there is a lot of data showing that. But the data is not as juicy as we would like to be, because this is very difficult. First, second, because and this is a bit of protest. Food companies try to sell you the message that food has a big effect on everything, but that is not true. We need food to survive. But if you want to put the effect of food on disease, for example, that effect cannot be big. Think about the biology. If you go on holiday for 2 months to some exotic, paradise where the food is completely different, will you get depressed? Not because of the food, right? Will your depression be cured? Hell no, right? So it's very important that we realize that we're receiving all this information telling us, oh, our food is good, our food is great, our food is fantastic, but the data does not support the big effects story. Effects are small. And this is what we have actually been able to publish through our different studies. We have been, big samples, UK Biobank, 350,000 people. The effect sizes are very small. We have smaller sample sizes, very, very well phenotyped, where we look at the relationship between diet and immune function and how the microbiota affects that. Effect size is very small. We also have family data from this Norwegian study called the Mother and Child study, the MOBA study, also over 100,000 participants, also very small effects. And we have shown different types of associations in terms of energy, in terms of association between sugary beverages like Coca-Cola. Please bleep that in the recording. So drinking Coca-Cola during pregnancy increases the number of ADHD symptoms in children at age 8. You do with that whatever you want. Mediation effects showing more causal relationships. So we've looked at different problems, and every time we find it as an effect, we find it small. So we're going to contemplate, we want to think that nutrition has an effect on neurodevelopment. We have to start from the fact that the effects are small. And this is very important because it changed the mindset of people. Oh, because food is so important. Yes, it is important. But for ADHD, the importance is relative to other things. The same for depression, the same for other. You could also apply it for neurodegeneration. Now, I make emphasis on this paper because I'm very proud because we actually created a clinical trial where we tested two diets, very hard diet, this restriction elimination diet, a healthy diet based on a Mediterranean diet, and treatment as usual. So treatment with pharmacology. And to keep the story very short, the results of after 5 weeks of treatment, treatment is a very big word, 5 weeks of diet, we found that the children and families with ADHD that actually followed the healthy diet, which is an easy diet to apply, had a better, a higher symptom reduction compared to this restriction elimination diet, which is very hard, very difficult, and almost as good as treatment as usual after 5 weeks. So it could be, chance finding, whatever. But the 1 year follow-up of this same group will come out published hopefully before the summer. We're revising the manuscript. And the whole point of this is that we are putting out there that diet for the first time can be seriously considered as sort of co-treatment for people with ADHD. And this is quite important for two reasons. When you're a child and you're diagnosed with ADHD, your parents take over control and they decide for you. And they tell you, you have to take the pills because these pills are good for you to some extent. Take the pills and you shut up and you do it, right? But when you're an adult, not anymore. You decide yourself, right? And adults with ADHD are epidemiologically at risk of everything bad you can think of. Death, overall death, divorce, going to jail, dying in a car crash, losing their jobs, getting addicted to substances, risky behavior. Right? The epidemiology is very clear. There's a nice review called Live Fast, Die Young, the story of adult ADHD. It was published, I think, shortly before COVID or during COVID. And they describe the whole epidemiological picture of somebody that has ADHD as an adult that does not use medication compared to the medication. Now, the patients, they complain. They say, I don't take the medication, not because I'm stupid, but because I don't like what happens with me when I take it. And you have to respect that. When you're treating people and working with them, you have to respect what they feel, how they change. So I'm not a psychiatrist, so I won't argue with that. But I would like to see if we could actually use diet to help people use the medication more, not more medication, but adhere more to the treatment. Perhaps using less amount of milligrams, I don't know, less frequent. There is something, I don't remember exactly the name, but maybe some of you knows that psychiatrists are now advising patients, use the medication when you feel you need it. And it can be applied to some disorders, not others, of course. But in the case of ADHD, seems to work. So putting all those things together, the whole concept of using nutrition to help people adhere more to treatment, at least for me, is a very good idea. Maybe it's not the nutrition at all. Maybe I have it all wrong. That's fine. Maybe it's the structure of eating at the same time every day, of cooking the food in this way. I don't know. But if it helps, fine. I'll find something else to do. Now, so I think that we can establish that this line, while not as strong as this one, is pointing towards something true, right? Now we want to see if we can find this link. Maybe the whole thing, this connection between diet and ADHD has to necessarily go like this. Maybe there's a direct effect. And this is where the neurotransmitters story that I told you comes into play, right? Now, there is quite a long growing evidence, and I'm just going to let it scroll because there are multiple things happening. The most important thing for me is that people are becoming more aware about how strict and systematic you need to be if you want to study this. This is not, it's not a game. You need to be serious about it, and you need to go beyond, the marvelous world of biology and actually put some systematic, some statistics, some bioinformatics, some method to it. Okay, not to say anything about biologists. I'm one of those, right? But the data is accumulating in such a way that the only thing that you can do is keep researching, keep going, keep going. Now, we have our approach. We've developed this. I told you I started many years ago, and when I started, there was nothing. I knew nothing about this. I was just interested, and I needed to do something in terms of research. So what as I told you before, I'm trying to bring all this systematicity from genetic epidemiology and statistical genetics to this world. So we use things like meta-analysis, which are pretty common in other research in clinical research, right? Meta-analysis is a pretty old method. So why not? It works very robustly everywhere, right? We're applying the RDoc approach. Are you familiar with RDoc? So RDoc was created in the US, I think in the late 90s or maybe earlier. It's basically to try to move beyond the clinical categories, so case, control, and actually use component phenotypes to create new groups. So for example, if you think about Alzheimer and Parkinson, right? Neurodegenerative disorders, which you could diagnose based on some particular criteria and then group people, put them in boxes. What if I told you, why don't you look at the brain size? Or why don't you look at this particular level of this particular marker? Or why don't you look at, some behavioral readout? So they stop becoming patient of Parkinson, a patient of Alzheimer, but actually having a number on this level of this protein, this number of cubic volume of their brain, right? So you start transforming their clinical outcome, their clinical diagnosis, into measurable, objective readouts that are shared. And this is the trick. They are shared across different groups. That perhaps on their own, they are not enough to diagnose, but they're strong enough that you can see them, you can see variants of these things in people with Parkinson's or people with Alzheimer or people with other neurodegenerative disorder. So you move away from the clinical outcome, and you go into the more objective quantitative measurements that can also have clinical relevance. The things that they are not used officially to diagnose. Is that clear, more or less? Okay. Now, we have a multivariate approach, trying to link variation in microbiota with variation in the brain. I'm not going to talk about that today. And we use animal models. So I'm going to mention these three, an example of those three. It's going to take a little bit of time, but I think we still have some time before I need to stop, and then you can ask the questions. So before I do that, I need to explain a couple of key concepts so you understand what we're talking about. So we're talking about diversity. We're talking about what is there and how much of it, okay, in terms of bacteria. There was a big discussion before about maybe it is what you have and what other people don't have, or the opposite. In my opinion, it's about how much you have. We all live more or less in the same place. We drink the same water. We eat more or less the same food, right? Even in your family, if somebody's affected with ADHD or depression, I mean, you share the same food, you live in the same house, you have the same pet. So why would the type of bacteria that live inside you be different between you two? That doesn't make sense. What makes sense is the amount is different. And the amount can be a consequence of how much you eat something, or if you move more or less, or if you live in the city or outside the city. But remember, this is coevolution. So it's not going to be like now you have bacteria group A and you have bacteria group B. That makes no sense. There is diversity, of course. And if you live in the south of Argentina and compare somebody with the north of Sweden, sure, you're going to find some big differences, but you're also going to find a lot of similarities, which is pretty cool, right? Now, so we can quantify those differences between groups. And there's something called alpha diversity, and this is the explanation, and you can read. So it's basically telling you how an individual differs from another. If you have more of something or less of something, right? And you can quantify it in terms of groups, or you can quantify it in saying this is more than that one. And there are different ways of estimating alpha diversity. Okay? So this is within an individual individual, sorry, you can count how abundant things are. Very simple. It's very simple measure, actually. And there are like 5 variations of Alzheimer's diversity, which look at slightly different things. Now you have beta diversity. This is a little bit more interesting, right? Because it compares groups. And it tells you, okay, the groups are very similar or they are not so similar, right? And you can also, but you need to be aware because while they could be similar or not, it depends on how similar they are. So the beta diversity is telling you, well, they are similar because they share a lot of things, so they have a high similarity, or they only share a few things, so there's a bit lower, right? So this is way more interesting because if you're looking at people with ADHD and people without, this is the kind of things you want to look at because it's telling you how similar they are, not how different they are. So they are less similar. It's not exactly the same, okay? Because this.
Okay. So we run a meta-analysis, and I will show you the actual samples we used. No, not that. Okay, so this is the sample size. So as you can see, the N is not really fantastic, but at the time we did this, there was nothing or almost nothing. So we were one of the pioneers in getting these things out. There are somewhat bigger samples now on ADHD. I don't think so. So we had three Dutch samples and one Spanish cohort, and then we combined them. And well, we have an interesting sample size. It's not fantastic, but it's not bad. And the whole hypothesis, we can actually test it with our limitations. And I'm very proud because it was a big effort, getting everything together, sequencing everything, getting the money to do it, you know. And we made a very nice picture of this diversity that we find. So there are, you know, specific things related to ADHD, and there are other things related to the microbiota. So which part of the gene we sequenced, right? And if you choose different parts of the gene to sequence, you will add noise. I think you can understand that. And of course, if we use different chemistry, you add noise. And remember, we're talking about a very sensitive tissue. We collect the samples from feces. So this is very, very delicate. So you need to process them very fast and take a lot of care for it. And if you don't do that, you have a big problem. This kind of problem. So this graph, this very arid slide you're seeing, shows, for me, shows a terrible, terrible problem, right? It's telling you that you have 3 groups. When you want to compare things and analyze them together, even if it's a meta-analysis, what you want is to see one group and finding differences within that group. You don't want 3 groups. It's a big problem, right? And we always put this slide, because this is real data, we always put this slide to show people that they need to be careful on how to handle this. And they need to report how things were sequenced. They need to report when the sample was collected. They need to report how it was processed and managed. And if they used a -20 freezer or a -80 freezer, et cetera, et cetera. You can adjust for this. You can adjust for this problem. And what you're seeing here is a result of the sequencing of the sample. These 2 samples here were sequenced in the same way. These 2 were sequenced in a different way. This is a sequence, what is called a sequence artifact, okay? So what you're seeing is that samples that are sequenced in the same way, they cluster together, and samples that are, and the same applies for every sequencing technology, which you can see here. So they were sequenced with different chemistry and a different region. And that diversity, that artifact, you can see it like that. So when you see this is a normal case control step you need to take with this data. You have to keep them separate. For any reason, combine them, because you're going to make a mistake. So you need to keep them separate. And that's why a meta-analysis is so useful here. Now, the second problem is that what you measure is very strange. Because what you do is you sequence a small part of the genome, right, of the bacteria. And then you put those results to a machine that actually tries to identify what sequences are there. And it will find a lot of weird things and chimeras results and things that don't make any sense. But then he will start to find a lot of things that make sense, that are actually the sequences you actually wanted to see. The thing is that not everyone has all of the sequences that you observe. What does that mean? That if I sequence all of us here, you will have some things that are only yours, that nobody else will have. So you will find them with her sample, and everybody else here will be a 0. Right? Clear? The other thing that can happen is that I have some things in me that are a little bit rare, right? But maybe they're a bit more rare in Pascual. So we cannot detect them properly. So while I have some number, some amount of that sequence, he will also be a 0. But his 0 is not the same as the other one. And that is a big problem in this type of data, what is called compositional data. Everything has to add up to 100. Right? So all the zeros are not the same. And we struggle with that a little bit. But you know, old statistics from the 70s save the day. And what you do is you basically transform your data, because you're not sure if you're a true 0 or a methodological 0 or a whatever type of 0. You don't know that. So you have to eat them. You know, eat them. And there's nothing around that. You can do some QC and whatever. But what you do then is you transform the data. There are several ways to transform the data, but this is the best one. What is called a centered log ratio. I won't explain how it is. You can Google it. You can transform the data and it becomes useful. The zeros stop becoming zeros. They stop giving, they don't give extra information, but there are numbers, non-zero numbers that then you can use in your calculations. And this is very good because then you can do your analysis. So 2 things, you need to check how your samples were produced, and you need to transform the data once you sequence. Otherwise, it's BS. And many people, guess what? They skip one of these things. And current publications, for example, are done without, transforming the data, whatever. Now, when we run our meta-analysis, we applied one of the things that is now considered the good research practice for this data, and it's that you should have two statistical methods to prove a point. So when you run the associations, you should do it with two types of methods, and hopefully you will find the same result. Okay, so that's what we did. So I'm very old school, very boring guy. So I like logistic regression. You have two groups, you do logistic regression. That's how I learned it. That's how it is, period. No argument, right? Of course, knowledge goes faster. Younger people know more than you. So my students said, no, but we have better methods. Okay, but we still do logistic regression. Yes, okay, whatever, right? So we ran the logistic regression and we found, I'm not gonna explain to you what they do, okay? So don't get excited. We can talk about it later. But we found some association. And this is corrected for, I think we started with, 200 genera, 200 bacteria groups. And we ended up with these 5. And these 5 will survive some sort of multiple testing correction. But this was only using logistic regression. They are not true until we run it through the other method. The other method is called ALDEX, right? And it's a method that actually reverts the regression. So in logistic regression, your Y is the count, 0, 1, your case and control, and your microbiota is the predictor. ALDEX puts it the other way around, right? But for me, that makes no sense because I'm interested in ADHD. I'm not interested in the variation of the microbiota per se. I'm interested in the disease, right? The disorder. I want to understand the disorder and if the microbiota is playing a role. But of course, that's one narrow vision of the problem, okay? So when we flip it around, the data needs to be corrected and transformed. It needs to be normally distributed and to follow all the nice rules of regression. But guess what? 4 out of the 5 came back, right? I mean, it's not surprising that you see something coming back. It's the same data, right? But you're modifying the data. You're twisting the even though the relationship don't change if you change things between the equation. The fact that you transform it and you treat the data differently makes an effect. And the statistics are different, slightly different between logistic regression and ALEC. So we were very happy that we could save 4 out of the 5. And then you take those 4 out of the 5, then you go to literature and you start looking to see what these things do. And then is when all the happiness of finding this goes to die. Because there's almost no information. So that this is basically like 20 years ago when we were doing candidate gene analyses, right? When you found a gene associated with your disease, your favorite disease, and you went back to the books, what is this gene doing? Nobody knew. So you had to come up with this wild interpretation, right? I hate that, by the way. I hate that. But the paper is written in a way where we say in a very polite way, we don't really know what is going on. There is some data pointing towards inflammation. It's not really sure what's going on. The point is that there is a signal. And we're putting it out there for other people to go and test it. So what we need now is other people to get their cohorts, collect the samples, do something similar like we did, and test it. And with some luck, they will find the same thing. And then we have more evidence. And then we can maybe convince the microbiologists to try to grow some of these bacteria. Because within this genus, you have tens, maybe hundreds of species. We don't know. This is just a natural data reduction step because this is a genus. So under genus, you have species. It could be hundreds. It could be, 1,000 species. Who knows, right? So again, this is the signal detection. Okay. Moving to the next one. Oh, well, it's not that. This is a more recent, bigger. I wanted to share this very quickly. This came in JAMA not so long ago, where they actually did the same, but in a cross-disorder approach. So we did it focusing on one disease. They looked at multiple diseases, trying to find, like a general picture of microbiota, which they did find, which is not surprising, but it's very good that you can see it. This is a pretty big sample. And interestingly, they actually are able to show some of the putative functions that could be involved in this. And then inflammation, short-chain fatty acid production, some differences between depression and bipolar compared to other diseases. They give a pretty interesting picture there. So if you're interested, you can look out for this paper, because I believe this is being recorded, so you can check it out. Okay. One other thing that we see in psychiatry is this very complicated comorbidity. So there is no pure ADHD patient. These people present with a lot of things: OCD, obsessive compulsive disorder, conduct disorder, dyslexia, depression, bipolar disorder, and that is across. People with depression also present with anxiety disorders, mood disorders, etc. You get the picture, right? You get what I'm trying to say, right? These things are genetically correlated. They are epidemiologically correlated. It is pretty impressive how these people can cope with so many problems. Now, the thing is that when you bring that down into a clinical categorization, then some problems arise, because you cannot be two things at the same time, right? You need a diagnosis that is well described, so a diagnosis of depression, and you get treatment for that. But what happens if you have ADHD comorbid with your depression? Then it becomes very complicated, because how do we avoid interactions? Can we treat with SSRIs and methylphenidate? Do we need a non-stimulant to get it? You get the picture, right? Now, this is how we, that's one of the motivations where we like this RDoC, this transdiagnostic perspective. When you can look at different clinical outcomes together by using some of these objective measures, right? And for that, we use this mindset study. It's something that we created in Nijmegen. It's a clinical cohort that was recruited and not that re-diagnosed, but quantitative measures were collected from these participants on top of their clinical diagnosis. So they very generously accepted to be quote unquote diagnosed using this approach. So like 6 hours of test batteries, MRIs, blood, feces, well, the works. Real amazing work. And then people with ADHD, autism, bipolar, major depression, and schizophrenia were invited to participate. Okay. Now, and the whole point of this particular part of the work, this is quite big, but what we are doing is we're looking at microbiota, and we find a microbiota signal that is transdiagnostic. Can we use the microbiota to say something about multiple diagnoses? Is this a unique thing? Is this a shared thing? Coming back to this meta-analysis that I showed you before. Now, this is a picture of the sample. So very briefly, these are the substance use disorder, anxiety, autism, ADHD, mood disorder, right? And this is the count. And then you can see with the line if they are comorbid. So there is the group of people that are supposedly pure, right? And these are the comorbidity distribution. As you can see, most of them are comorbid with something. So it's a very interesting. This is a very interesting sample. These are that is the end of the sample size with gut microbiota collected. The total sample is a little bit bigger. Yeah, it's pretty cool, I think. Pretty unique. These are the pure samples, the comorbids, and this is what was measured. These are the full questionnaires, plus all the sub-questionnaires and sub-scales that are included there. This is pretty extensive, right? And what we did is that we joined them, we combined them using what is called a principal component analysis. You probably know about it, in order to do data reduction. That's basically what you do PCA. You simplify the complexity of your data, and you do data reduction with principal component analysis. And this is based on work that other colleagues using this data did. And we come up with 4 domains. And these 4 domains basically include all the information of all these questionnaires. These questionnaires were completed by all the patients that I just showed you. These 4 domains indicate some negative valence, some ability to have social processes, cognitive performances, and some arousal and regulation issues. And this might sound familiar to some of you because they say something immediately about what type of disease they could be, right? And the idea is that these things resemble these RDoC domains, which are built based upon these shared quantitative readouts that you can collect from the sample. Now, the whole premise of the work is to relate if we can see variability in the domains that links with the variability of the gut microbiome. Okay, clear so far? Now, this is the analysis strategy. So we use a sort of package that does regression, right? But before we do that, we apply what is called a randomized lasso, which is basically a way to select. So instead of doing, instead of testing everything, we select those markers, those bacterial markers that seem to be better, that's showing some sort of degree of variance with our outcome. Again, I'm not going to go into detail, but this is basically a step that we use to reduce the data complexity from the microbiota side. Okay? The previous one, this is done to reduce the complexity from the behavioral side. And this is done to reduce the complexity from the bacterial analysis. Okay? Now, the model that we use to correct our regression, and of course, we try to do some interpretation once we do our analysis. Now, when we run the regression versus the functional domains, we see a sort of distribution of associations. These are the positive associated, significantly associated after correction, right? In terms of the direction. So for the negative valence, these are the bacteria, and you see if you have more or less. So the higher amount, the higher score in the domain. And if you score higher, that's worse. Okay? So this positive correlation is basically telling you the more you have this bacteria, the worse it is for you in terms of those readouts that we created. More or less clear? Okay. Now, the same for social process. Here we found 5. Here we found 1. In cognitive system, we found 2 with different directions. So again, another layer of complexity because we don't know what is the direction of this. Is having more worse for you? Is having less worse for you? Well, both actually, because this is a biological system that is redundant and things change. So that's why we need these kind of approaches so we can catalog the distribution of effects a bit better. I mean, making a hypothesis here is ridiculous because we don't know. Now, and the last one, the arousal, also we only found 1. These are after correction. So this is highly, highly corrected in terms of statistics, multiple testing and all kind of things. So if they survive, is because they seem to be really there. Okay? Now, the second thing we did is this driven by the patients? Is this an artifact of the difference between the patients and controls or something like that? When we remove the controls from the analysis, we see that the behavior of the associations basically stays more or less the same, telling you that this is an effect that is coming from the real difference between patients and controls. So it's not a control explained effect. It's not the healthy people explaining the association. Okay? It's the affected people, which is what you want to see, right? Now, when we actually, this is very nice and very organized, right? You have your domain, and then you have some associations with some directionality, right? Then you have some of these bacteria that are shared between 2 domains, right? So it's very nice and organized. But when you put it in terms of the actual clinical outcomes, which are the ones that these things were based, well, this is what you see. Disorder, chaos, right? Which is what you expect to see in real life. Because these groups, well, clinically make a lot of sense, to organize people, biologically, they don't need to make any sense for the bacteria. The bacteria does not recognize schizophrenia or autism. Doesn't work like that. So the idea of using a RDoC approach reduces a little bit this chaotic effect. I'm not saying it's perfect. I'm not saying this is bad. I'm just saying that it's a very useful tool that you can apply and combine with the usually used clinical outcomes. Okay? Especially in samples that have a lot of comorbidity. Okay? So again, we try to make a functional interpretation, and because, yeah, we need to try to explain what is going on. We found things like, availability of nutrients. We look at pH. Community composition is very difficult to do because we cannot see very well how these communities interact in the gut. It's a very difficult thing to do. One of the things that we found once you look at the different bacteria across the different domains is that there is a higher incidence of bacteria that are involved in inflammation or pro-inflammatory processes. It could be just a fact, it could just be a result of the fact that the microbiota and our immune function are so highly intertwined that we cannot split them out. And we see what the only things we can see. But inflammatory processes keep coming and coming and coming. I don't know exactly what it means yet, but this is what we're seeing. Now, the opposite is seen when you have a lower abundance, but yeah, again, it could be just a matter of looking for the keys where there is light. You know that comment, right? You lose your keys in a dark room, and then suddenly a light appears, and then you go there to search for your keys, and then your friend asks you, "Why are you looking there? Are your keys there?" You say no, but here's where there is light. So this is the problem we're facing, right? We're looking where there is light, and that is fine. It's incomplete, but it's still fine, right? And I will end here because I think everybody's hungry. We wanted to explore, and this was based on a paper published in Cell, I think in 2019 or 18, where they transplanted the feces of an obese man to a mouse, and the mouse became obese. And they transplanted the feces of the twin brother of the obese man who was lean into the mouse, and the mouse went back to its original weight. And I read the paper, and I thought, this is black magic. No. So I went to a colleague of mine, and I said to her, I want to do this with ADHD. I want to give ADHD to mice, right? So we recruited patients and controls, and we transplanted feces to mice, to bulb mice, the control mice, right? We didn't give them ADHD. Okay, let's start from there. But we really messed them up in a way that I was not expecting. So first of all, we were able to track back who was the case and who was not based on the bacterial distribution that we saw in the mice. Once we transplanted it, we went back to the animals, collected their feces, sequenced that, and we were able to track back who were the cases and who were the control, which I was not expecting at all. Like showing that there is this clear disease effect, let's call it like that, right? That you can even track back after the transplantation. This was really unexpected. I thought that we were gonna see one big blob, okay, because of what I told you. But with putting all together the other findings of the other study and this, I think it becomes a little bit more easy to see that perhaps we can map these bacteria differences or in abundance a little bit easier. Now, in terms of behavior, again, we didn't give them ADHD, but they were really, really developed in a different way. Those animals that received feces from people with ADHD versus people that received feces from controls. And they were, more anxious. They moved less. They were more fearful. These are all hallmarks of ADHD, not diagnostic ones, but they're really behavioral hallmarks. So I don't want to extend too much. What does it mean that an animal is fearful for a human? Or what are the interpretations of that? Because that's not how it works. But the point I want to make is that why are they different? They were reared in isolated cages alone. All of them had the same type of chow, the same type of food, same type of water. The only thing that changed was they were transplanted with different types of feces. The oral gavage was given by the same person at the same time of day. Why are they different? It's the bacteria, but why, right? So there is an effect, there is a functional effect where it's just, we need to discover. Okay. We're doing a lot of things. We're a very small group with a little bit of money, and we're doing a lot of things. We're very stretched out, trying to, push these hypotheses, these ideas, right? We want to work in more aspects of central nervous system, brain capacity, brain volume, brain connectivity. These things are very important for us also. We want to understand if inflammation is actually something that we should be looking at more closely for neurodevelopment. I'm not a big fan of chronic inflammation and neurodevelopment, these kind of things. I think it's too not specific, but it might just be the one. I don't know yet. Can we actually modify the kinetics of neurotransmitters in the brain with nutrition? If that happens, big party, to say the least, right? Can we do that by identifying a sort of signaling, an ADHD signal? So far, the evidence says no, there is no disease-related signal, at least for neurodevelopment. So perhaps it's a good idea to pull them all together and combine them. Yeah. Can we use this, for example, to treat with diet? I think that we need to work more on it, but I think nutrition could be a very good avenue for this. Transcending neurodevelopment could be psychiatric, even neurodegeneration. I don't believe in probiotics for this, personally. I've tested them. I've helped work with them, and I don't think they work for this at all, for brain health, let's say. But specific diet might do the trick, because it's a completely different way of working. There are other things that you can think of in terms of disease. Can you think about treatment, for example? Why people respond and others don't, if they have the same profile, let's say. Epigenetics, this is a clear textbook gene-environment interaction. So it's epigenetics to the core. Could you use microbiota as a screening tool to identify people at higher risk, for example, together with questionnaires? Imagine a mini mental plus microbiota. Do you have somebody that is a higher risk of Alzheimer's? That it's completely doable, completely testable, right? And of course, you want to learn a little bit of the biology. You can replace ADHD at the bottom with any other thing, and you can learn a little bit about the biology. There's a couple of groups in the Netherlands that are working a lot of that. They do what I call trait analysis. They investigate traits, so behavior or brain volume or some level of some protein or whatever. Not necessarily a disease-related trait, but trait. So they can learn more about the biology, which is not my cup of tea, but it's as valid as anything else, anything that I do. Good for them, I guess. And this is the team. This is the big team. There's a lot of people that we work with. Joanna, Babette, Priscilla, Miriam, and Danik, they are part of my direct team. This is the group from Valdebron in Barcelona. This is our team from this team here from Radboud. Yeah, psychiatry department. We got some money from the EU to do part of this. You can check it out. The website is New Brain Nutrition. The website is still working, it's still active, being updated. We give actual recommendations in terms of food based on the findings we got during our research. You can read about the research. The cohort is called Eat to Be Nice. It's a European project that I coordinate. It's finished now, but the resource is active, it's working. And we have, I don't have an example, but we have videos based, this is data, this is evidence-based all of this is evidence-based, okay? That's it. Thank you very much. Happy to take questions if there's time.
Thank you very much for this fantastic presentation, as usual. Questions from for Alejo? Alberto?
Well, thank you. Thank you very much for, it's very evoking and very inspiring. But I was just wondering about age. Is there, what is underlying the changes on microbiota with age? Does it? I was just doing some imagination. Is aging of the of the person, of the host, or does the system ages? Does the whole ecology of the within the gut age? Is there something like that? So does it evolve with time?
Well, the system ages, right? Because we are the system. So our gut is the system. We age. But aging is not only that we, let's say, are less efficient in our metabolic machinery, like our cells divide a little bit sloppier, there are copies in our DNA come with more errors, etc., which is aging. But also that our environmental patterns are different. So you eat different, right? The frequency of things you eat changes. The type of stress you feel is different. The amount of time you're exposed to that stress changes with age. Life events, which is something I didn't talk about, they accumulate with time. So, happy life events, traumatic life events, they also accumulate with time. So yeah, I think it's a combination of the system being older, so the biology not functioning as such, but also that our brain is getting different types of stimuli, which subsequently communicate in a different way with the gut. You can see a very extreme example of that is babies. We have a cohort of babies, and you can see the microbiota jumping from the time they eat breast milk when solid food is introduced. The microbiota changes, like 2 groups. The same person, you see, that is an extreme age effect produced by diet, but it's the same for others, right? The use of alcohol, for example, you can also see in the microbiota. So around age 20 or something, people start really drinking, then you can see a shift in the microbiota. That is also aging to some extent.
Questions?
First, one comment, general comment. That seems to me very logical that feces and blood are related brain into brain disorder. This from an Aristotelian point of view, very logical.
Correct.
Then 2 questions. First of all, really, I was fascinating because I thought that a hyperactive child has some genetic background, but put a slide with 8% of heritability. It's a very high figure. Yeah. What is the action? Only 20% to modify this disorder? Only 20% with the diet, physical exercise, drug, and so on.
Yeah.
This is true.
Well, it is partially true. It's not any. I wouldn't interpret it like that, because the heritability includes everything genetic
and it includes the interactions with the environment. So the way that the formula works is that it takes all what is called the additive genetics. This additive genetics includes the interaction with the environment. And we cannot separate it in the way the heritabilities are calculated here. There are other ways to estimate heritability. That is called the SNP-based heritability. And it only takes the single nucleotide changes in the sequence and accounts for how much of the variance is explained by these changes in the sequence. So it's purely common genetic variation. And for ADHD, I think it's around 22%. So it completely shifts. So the heritability, what is called twin-based, which is what I showed,
Yes.
it's around 75%. When you do only solely common genetic variants.
Excuse me, but 75% is a very big one.
Sure.
In twin, but you know the twin studies are in some way false because it's not only genetic, it's the twin have genetic but epigenetic,
Sure.
diet, environment.
And those interactions are included there.
Are included.
Yes,
Right,
correct.
you include the major part of the conduct, the really.
Well, the interaction between genes and so are included in this twin-based heritability. So it could be epigenetic mechanisms, which is our interaction.
Yes, of course.
And others, which could be the gut bacteria. We don't know. But if you want to say separate the genetic plus and the non-genetic, the fact that the proportion is smaller is not telling you anything about the effect. So you're explaining variation, right? But the size of the signal doesn't correlate with that amount of variance that it is missing, let's say. So we say with 25% is missing, right? Or it's non-genetic. But the effect of those non-genetic factors can be bigger. That is how you should look at it.
Well, the second one, more complex. I have no experience with the child hyperactivity by the chief of my hospital. It's a big hospital, Dr. Mateus, told me always that the increasing and the rise of consultation of this disorder may be related to social aspect. That is to say, the TB on child, the phones on child change the activity, the physical activity, the group interaction of the child, of the children. For that reason, I think that in this disorder, the social aspect must have a more important role that the general medicine discuss at the moment.
That's a very important point. The social aspect of neurodevelopmental disorders have been highly discussed. So there are a couple of things to know. So a higher frequency of consultation does not correlate with the incidence. But it's important. It's important if you don't know about the clinics of it. So more consultations mean more disease. Right? So that's one. Now, social aspects were very nicely investigated in a meta-analysis published, I think, in 2017, in the American Journal of Psychiatry, where they investigated time in front of the TV. We didn't have mobile phones like we have now. Still, time in front of the screen, food with food additives and colorants. There were like 4 or 5 really social environmental aspects. And none of them correlated. Food additives was the only one that more or less was in. The rest, none of them, negative. A very nice meta-analysis showing that on their own, social exposures are not enough to trigger ADHD. They might exacerbate some behaviors, but these behaviors might go away once the trigger is closed. In ADHD, the behavior doesn't go away. They don't go. So the hyperactivity, it doesn't, right? The lack of attention, until you find a thing, one thing, the lack of attention, the shifting does not stop, right? So that is also very important to me.
there is what I mean, Mendel described as sort of innate. Right? People that are alike reproduce. They come together and reproduce. And we find that also for ADHD. So people with ADHD tend to get together with other people with ADHD. So that masks some, because some of them might be more extreme than others, right? And then that could mask some sort of parental problem, because it was thought a few years ago that bad parents cause ADHD, which was a big issue in psychiatry because people were not getting the treatment. Parents were feeling like terrible, you know, I'm destroying my child, you know, it's very, very bad things, right? Very bad, because you're blaming yourself for something that you did. A lot of interest was placed in trying to describe the behavior of parents of children with ADHD. And of course, big surprise, they are also, but nobody knew, right? So the higher number of consultations of adults is also to become more aware that we don't only need to look at the children, we also should look at parents. And in more cases than not, there is a need in that that is measured. I will not disagree that the social aspect is very important. It is, because the pressure it exerts in somebody's mobile at the time. If you don't have ADHD, you can switch very easily. Put down and move on. But if you have ADHD, you might be more difficult to switch. So you become more addicted, not addicted, but you become more reliant on the screen to function. See the problem? So this ability to switch is less. Now ADHD, we all suffer from what is called this delay aversion. So normally for some things we like a reward very fastly. So if I tell you, do you want €5 now or €20 in a week, which one will you choose? 5 now or 20 in a week? 20 in a week, right? In general, people take 20 in a week. In ADHD, it's the opposite. In general, they take the shorter, and social pressures stimulate that. Screen time, TV, etc. They are designed to push this delay aversion to the extreme. So if you're susceptible, maybe we should screen you for one of them. Thank you.
Okay, I think the last question. I just to bring your topic to our field. I mean, it came to my mind many, many hypotheses that we could test. I mean, this microbiota could probably influence behavioral symptoms, the progression of the patients, but we've been discussing these days, no? The interpretation, now that I see your presentation, it's really complex. I mean, I think it's diabolic, actually, sometimes. I mean, yeah. I mean, because we are talking about, many interactions, as you beautifully show, that could be related to etiology, maybe, probably not, because, or yes, I mean, it depends. More.
I mean, we believe
Yeah.
even though they have a small effect, but we believe that they have
Yeah.
So effect size is not
Yeah. But could be also reverse causation. Could be that you start eating differently depending on all these. Definitely. And it could be also what you're saying, which is a really beautiful idea, that there could be a not enough compensation, and then you see the symptoms going out. All these different hypotheses could be part of the explanation, right? So the Wild West, as you said.
For Alzheimer, it's complicated.
very much, Alejo. We have to stop here. Thank you very much.